guys, today's video is about enzymes, which are the first topic in our next unit, which kind of focuses on how cells both use and produce energy or like convert energy into other forms. So starting with enzymes, and then we're going to move from here into photosynthesis and cellular respiration for the rest of so with that, a lot of this stuff you might remember from your freshman biology class about enzymes. And then, of course, since we're in AP Bio now, we're going to add on a little more complexity to what you might have learned before. So here we go. All right. So before we talk about enzymes, it's important for us to sort of define what metabolism means, which is a word that probably you hear thrown around a lot, or maybe you throw it around. A lot of times you'll hear people say things like, I have a fast metabolism, or I have a slow metabolism, or whatever. And they're usually referring to sort of how fast their food breaks down. And also a lot of times people will like use it in relation to like weight loss or gain, which it can be, but metabolism is actually a lot more than that. So it is, your metabolism is all the chemical reactions happening in your body, sort of like combined. And there's two different versions of reactions that could be happening in your body and are happening in your body all the time. So you have catabolic reactions and you have anabolic reactions. And catabolic reactions are going to be breakdown pathways. So that's things like, an example is digestion. So a lot of the enzymes that we like commonly think of are digestive enzymes that are related to catabolism, so breakdown and digestion of nutrients into smaller, more usable parts. But there are also anabolic pathways, and they're part of your metabolism too. So those are synthesis. So building things up. An example of this would be like building muscle. Building muscle. It could also be things like assembling new protein units from amino acids. It could be making copies of your DNA so that your cells can go through mitosis because it has to have a new set of DNA. So anything that's related to sort of build up is going to be an anabolic pathway, and that's the difference. What's responsible for this is all these different enzymes that are responsible for adding things together and also breaking things apart. So enzymes are proteins, which we'll talk about that more in a few minutes, but I'm just going to make a note that enzymes are always proteins, and they're going to be responsible for sort of like moving things through the steps of these processes. So a lot of them have like multiple steps, and that will require multiple different enzymes because, as we'll talk about, enzymes are super specific. Okay, cool. That's metabolism. Related to this also is the concept of free energy. So it is the portion of the energy that is able to perform work. There are two possible types of reactions that are going to be related to free energy, and they're exergonic and endergonic. Exo or exer is going to be related to letting things go or releasing them. So in an exergonic reaction, what you're experiencing is free energy being released. And in an endergonic reaction, that energy is going to be absorbed. Sometimes this is also sort of related to heat being absorbed and released. And those are, that's a concept that you may have heard of before, that some reactions are going to release heat and other reactions are going to absorb heat. And that's very closely connected to this concept of like free energy being absorbed and released as well. When it's related to heat, it'll be thermic, so exothermic and endothermic. And this is just very similar, but related to just free energy in general and not necessarily heat energy. Also really important for us before we get into enzymes to talk about ATP, which is the molecule that your cells use as free energy. ATP stands for adenosine triphosphate, which you can see here. It's called that because it has three phosphate groups, an adenine and a sugar. So if I label this, these three right here are phosphate groups. If you remember back to your functional groups from before, from biochem, that's what they are. And then you also have this sugar, and this is adenine, which is actually a nitrogenous base. So the pieces adenine and ribose, which is the sugar, are actually also used to build up RNA, as well as ATP. So what happens is the final bond between the second and the third phosphate, so this bond right here, is a super high energy bond. I'm going to actually zoom into this, which is where the energy is stored. So what happens when it's time for the energy to be released is the third phosphate is broken off, which you can see happening here. It's broken, and it produces ADP, which is adenosine diphosphate. So tri is 3 and di is 2. It's related to the number of phosphates. And then because of that bond was broken, the energy that was stored in the bond is also released and now is a free energy source that the cell can use to help do reactions. So this is also being done by hydrolysis, which you can see as the water is being inserted. So just like a polymer would be broken down, the same sort of concept there. And then also important just to note that this is a cycle. So ATP can be broken into ADP, and then later on it can be reformed. I'm just going to put arrows here. Back into storage when energy is available to be stored. So it's happening all the time in a cycle, over and over and over. Here's the molecular breakdown of ATP. Just if you're into the biochem, you can see that there are phosphate groups, and they're groups. They're not just like phosphate atoms. They are functional groups. And then you also have your adenine and your ribose there as well. So that's ATP. Here is just another version of that cycle that I was just talking about. So when you have ATP, you use hydrolysis to break it down. Energy is released. So that's what's happening over here. So when the energy is released, you have the ability to use it for an endergonic process, which means that energy would be reabsorbed in the next process. And then you can reform ATP by adding a phosphate to ADP, and that will store energy that came from a catabolic process that released the energy, and now it's able to be stored. So this is happening all the time in a cycle over and over and over again, in all of the different cells of your body. All right, also related to this energy conversation is the concept of activation energy, which is just the amount of energy that a reaction needs to begin. A lot of times this energy is going to be in the form of heat. For so many reactions, they require so much energy to get started that they actually won't ever happen without the help of an enzyme. So this is why enzymes are super important for making sure that you are able to actually stay alive, is that the reactions wouldn't even begin, or they would happen way, 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 way too slowly to actually support your survival. So activation energy, again, is just the amount of energy that's needed for the reaction to start. And what's going to happen with enzymes is that they're going to lower activation energy so that less energy is needed to actually make the reaction begin and happen. So I'm just going to make a note of that this means less energy is required to begin the reaction when you use an enzyme. Um, and that allows the reactions that your body needs to do, both anabolic and catabolic, to actually get off the ground and happen. When you look at this graph, the red line is the activation energy if you do not have an enzyme, and the black line is the activation energy with the enzyme. And then just another thing to note is that G, capital G, is representative of free energy. That's the variable that you use. So if you ever see that, it is the energy, free energy. Cool. All right. So now let's talk actually about enzymes and the basics of them, which probably this is the part that's going to be like, oh yeah, I know what this is and I've heard it before. We often refer to enzymes as catalysts. Catalysts are things that speed up reactions and allow them to happen faster. This is really happening because of that lowering of the activation energy that I talked about on the last slide. So catalyst is just something that speeds up the reaction. Enzymes are always specific to their substrates, which is what they're going to be breaking down. The label this is, or putting together. So this is what is modified by the enzyme. Um, and they will only match to that one specific substrate. So sort of like a lock and key type of ideal. And this goes back to talking about protein shapes and why it's so important that they are specific and folded correctly, because if they're not, they won't be able to do their job. And a great example of that is enzymes that won't be able to even bind to the substrate if they are not folded the correct way. And then I'll just also, this says this, they're not consumed in the reaction, which is true. So because of that, we call them reusable. So the substrate is going to be converted into a product or several products. The enzyme will be the same, completely unmodified by the reaction happening, and it will be able to be used over and over and over again, unless something terrible happens and it becomes denatured, which we'll talk about in a few minutes. So I want to sort of talk about this diagram and kind of use it as an example. Let's say here that our enzyme is lactase. You might remember that enzymes usually will end with ase and in ase. And that's one way that you can automatically be like, oh, that's an enzyme, which means it's a protein. The substrate doesn't have to be a protein. It could be really anything. In this example, I'm going to say that it is lactose. Sugars often end in ose, which we talked about a little bit when we did biochem. But this is a carb. It's a disaccharide. It's made up of a glucose and a galactose, actually. And it's, I'm sure you know this, but the sugar that's found in milk. So what will happen is the enzyme lactase and the substrate lactose are perfectly complementary to one another, like a lock and a key made to be together. So what will happen is the lactose will bind to the lactase at a very specific location, which is called the active site, which is where the reaction actually will occur. So it's right here. This red line represents the active site. When they're bound together, they're called the enzyme substrate complex. And then at the end of the reaction, the products will be released, which in this case is two sugars. They're not the right shapes, but I'll just label this one as glucose and this one as galactose. And then you can see here also that lactase remains the same as it did at the beginning. So one thing to note, and like a common issue with this, is that the enzyme is not always at the bottom of the picture, and it's not always the bigger item either. It is just the item that will remain unchanged throughout the entire reaction. The substrate should be different than it was at the beginning, should be the products now, which means it will look different if there's a diagram. So when you're trying to identify the enzyme, look for a molecule shape that has not changed as a result of the reaction. And there you go. So let's keep it moving. Okay, this might also be familiar to you that every enzyme works best at specific temperatures and pHs, and we call that the optimal or optimum conditions. So this means like best or ideal. And every enzyme, depending on where it is used in the body of a animal or plant, is going to have different optimal conditions. So we're going to look at these two graphs. The first one with the yellow background is related to pH. So you will always have reaction rate on the y-axis and then a different variable, which will usually always be temperature or pH on the x-axis. And then in this top graph, you can see that there are three different enzymes. They are pepsin, which is an enzyme that breaks down proteins in your stomach, salivary amylase, which is an enzyme that breaks down starch, and that's found in your mouth, and then arginase, which I actually don't know what arginase breaks down, but probably arginine, which is an amino acid. So what you see is that different enzymes have different pHs, which makes or different optimal pHs. So wherever the graph peaks, that is the optimal condition. So right here for pepsin, I would say that the optimum pH, oops, I don't know why I did that. pH is about 2, which is very acidic, of course. And that makes sense because pepsin is found in the stomach, which is very acidic. So the environment where the enzyme is meant to function is going to impact what its optimum conditions are. For salivary amylase, the optimum conditions are pretty close to neutral, so about 7. Optimum equals 7, and that makes sense because salivary amylase is found in your saliva. So we'll just say found in mouth, which is going to be a lot less acidic than your stomach is. If we look down here, this is the same sort of situation for temperature. So you can see two lines happening here. One is a typical human enzyme, which functions best at human body temperature. And then the other is a heat-loving bacteria that functions, their enzymes function best at about 77 degrees Celsius, which is quite hot. The point where the line starts to drop off, it's happening because the enzyme is becoming denatured, and that's occurring because it's too hot. So I'll just make a note here that enzyme loses function because it is denatured. And what denatured means is that shape is altered to no longer fit the substrate. So when you go back to thinking about like biochemistry and protein folding, all of that protein folding is just because of chemical bonds. And when you start to raise the temperature of things, the atoms start to move faster and the bonds start to break. And that causes like all that carefully folded, perfect protein shape just to sort of unravel. And as it unravels, it no longer can bind to the substrate or it changes properties altogether. So, for example, like an egg protein is clear when it's folded properly, but when it's heated and you cook an egg, it denatures and that causes the properties to change and it becomes opaque and white. And that's the egg whites. So, protein shape has a huge impact on the enzyme's ability to function. Okay. So that takes me right into talking about denaturing. There are tons of different factors that can affect a protein's ability to fold properly. Two that we just talked about were pH and temperature, but there are also others. So protein shape is determined by the individual's DNA, genetic code. So if there is a change in the genetic code that alters the sequence of amino acids, that primary structure, then you will have an incorrectly folded protein from the beginning. An example of that when it comes to enzymes, let me take my highlighter off. So I'll say example is lactose intolerance. So people who have lactose intolerant from the beginning of their lives, they have a different sequence of nucleotides in their DNA that's coding for a different sequence of amino acids, which means that they don't have lactase to break down lactose. And that's an example of a mutation sort of affecting an enzyme. pH, we already talked about. Chemicals can cause unfolding of a protein. Temperature, of course, we just talked about that. That's why it's super dangerous to have a really high fever for a long time, because your enzymes and other proteins that are really important can start to actually denature if you have a super high fever. Um, Also, the presence of something called a chaperonin can make a difference. So, a chaperonin is a structure that helps proteins fold correctly. You can almost think about them as like a tent that goes around like a newly folded protein to kind of like keep the environment and the conditions at their ideal levels. So if you usually have a chaperonin and then all of a sudden you don't have one anymore, that can cause a big difference for the protein's ability to fold correctly. And then also there are certain diseases that are really closely related to protein folding as well. And some of them are Alzheimer's, Parkinson's, and mad cow disease, which mad cow disease is a prion, which is super crazy protein folding story where the protein is folded incorrectly and then it actually like triggers other brain proteins to fold incorrectly too. They're not technically like living because they're proteins, so super crazy, crazier than a virus. Prion stuff is nuts. But certain diseases can affect protein's ability to fold as well. So denaturing or denaturation is unfolding. Sometimes you can have a situation where like once the temperature or the pH restabilizes, you can have a protein renature and fold again correctly. But typically we say denaturation or denaturing is bad news because the protein will not function. It's, I'm just going to say it over here. No function. Bad phase. So on those optimum graphs that we looked at on the last slide, this would be like your enzyme function is at zero. As it's decreasing, that denaturing process is occurring. So it's like slowly unfolding. Okay. So now with enzymes, there are certain molecules that can affect their abilities to function. So they also can be used to turn enzymes on and off. This is important because you might be thinking like, oh, I want my enzymes to all be turned on all the time. No, you don't. Enzymes have certain times that they need to be active and certain times that they need to be inactive. So it's important that there are different control molecules that are able to kind of be the on and off switch. And those are called cofactors and inhibitors. Cofactors are going to be helpers. So they're going to be used to turn enzymes. And usually the situation with that is that the enzyme is not in the correct shape to bind with the substrate normally. When the cofactor attaches to the enzyme, it changes the properties of the protein so that the active site becomes the correct shape. Now the substrate can bind. One thing just to note is that if the cofactor is organic, which remember organic means that it contains carbon, then instead of being called a cofactor, it's called a coenzyme. So if the cofactor is another protein, if it's a sugar, um, it would be a coenzyme and not a cofactor. But they're both helpers and they will be used to alter the shape of the enzyme so that the active site is the correct shape. So I'll just say that these are going to alter protein fold to make active site correct shape. So on the flip side of that is an inhibitor. Inhibitors are going to be used to turn off enzymes or inhibit the function, which means stop it. So over here, I'm going to say that inhibitors turn off enzymes. And they're going to block the substrate from being able to bind, either by literally binding where the substrate is supposed to bind, or by changing the shape, sort of like a cofactor would do, but changing it in a way that will not allow the substrate to bind. There's two different types of inhibitors. I'm going to draw my arrow to this picture and zoom into it. So you have either a competitive inhibitor, which means the inhibitor is going to block the substrate from binding. The active site is still the right shape, but the inhibitor sits right in the active site and prevents binding. So this is, I'll say molecule sits in active site and blocks it. So it's called a competitive because it's competing with the substrate for the binding site. A lot of toxins actually work this way. They'll bind where something else was supposed to bind. Hemoglobin is not an enzyme, but it's an example of something that like experiences competition with actually carbon monoxide, supposed to bind to oxygen gas, O2, to carry it around. But that's what happens with carbon monoxide poisoning, is that the carbon monoxide is the right shape to bind with the hemoglobin. And that blocks oxygen from being able to bind, which is why you can die from carbon monoxide poisoning. So that's an example of competition. And then the other option is that you could have a non-competitive inhibitor, which is going to bind, say, molecule binds elsewhere on enzyme and alters shape. So substrate doesn't fit. And this would be sort of like the opposite of a cofactor. And that is a non-competitive inhibitor because it's not competing with the substrate, it's just making it so that the substrate can't attach. Both of these would cause an enzyme to not be able to function and would turn off enzyme function. All right. Last thing I want to talk about is allosteric regulation. So sometimes you have groups of enzymes that are combined together to create sort of like a giant enzyme that's made of many subunits. And what will happen is either an activator or an inhibitor will bind to the subunit and cause changes. They will not bind where the active site is typically. Actually, they will never. The only time that they will bind to the active site is when there's like cooperation, but the activators and the inhibitors will not bind to the active sites. And they're going to cause the change to happen in all of the subunits of the enzyme. So allosteric regulation, I'm going to say only happens in subunit and here is an example. So when it's active, you can see that the active site is sort of the like indentation of the flower shape that's happening. There's 4 subunits. The regulatory sites are in between the different subunits. So the activator comes in, binds, and that stabilizes the active form so that the enzyme will be turned on. All of the subunits will be turned on together. Without the activator, the enzyme will kind of like shift back and forth between the active form, which is the top, and the inactive form, which is the bottom. And the bottom one is not able to bind to the substrate because it's the wrong shape. So an inhibitor can bind in that same like regulatory site or one of the other ones because there's 4 of them. Like this is a regulatory site, this is a regulatory site, this is a regulatory site, and this is a regulatory site. And that will stabilize the inactive form. So if the enzyme needed to be turned off for a long period of time, the inhibitor would stay attached to keep all the subunits from becoming active. For these allosteric situations, you also can experience cooperativity, which means that when one of the subunits binds to its substrate, all the other ones become like prepared to also bind to their substrates. So basically, it's like peer pressure where one is ready to work, so the rest are ready to work, kind of like triggered by the binding of the substrate to one of the subunits. So just keep in mind here that these allosteric situations only will occur when you have an enzyme that's multiple subunits. So it has that like quaternary protein folding situation going on. If not, it's not going to be allosteric. It's going to be either a cofactor or just a regular like competitive or non-competitive inhibitor. And then just to finish up, this is kind of like the next level of this, which we'll continue talking about as we go on to more and more stuff. But there are ways that your body can use feedback inhibition to kind of like keep these pathways moving or to say like, okay, we have enough now, we're going to like shut ourselves off. So that's what's happening here is that at the top you have a substrate, which is threonine, it's an amino acid that binds to a specific enzyme and it creates all of these different like products, which this is usually how it works. It's not like lactose is an easy example because it's like one and done, one enzyme, and then we're like digested. But for most things, there's multiple pathways, or I'm sorry, multiple enzymes within the pathway that are responsible for doing like intermediate modification of these things. And then finally, you get to the end product. So in this pathway, you have 5 enzymes that are responsible for getting you to the end product, which is this other amino acid isoleucine. So then what happens is like once there's enough isoleucine, the cell actually uses isoleucine as an inhibitor to turn off the enzyme that starts the whole process. So it's like, okay, we've got enough. We're going to turn off the pathway now using the actual product that we made, which is pretty cool. And then once the cell has used up all of that product, it will stop being a competitive inhibitor, I'm sorry, a non-competitive inhibitor, so that the pathway can turn back on and create more isoleucine. So basically, using the product as an inhibitor causes the cell to not like overproduce a product, which is amazing. And we'll talk more about feedback inhibition later on in this unit and in this year. So that's it for today, kind of overview of enzymes. Thanks for watching and see you in class.